In our research, we're stimulating neurons in the thalamus, specifically the ventrolateral ventroanterior thalamus. We're doing this firstly because we know that there are strong projections from this particular area of the thalamus to the motor cortex that controls voluntary movement. So our hypothesis is that, and we have early data to support this, is that if we stimulate these cells in the ventral thalamus, they promote movement via an excitatory connection with cells in the motor cortex that connect directly to the motor neurons that then connect to the muscles. So what we're really hoping to achieve with this project is we want to show that optogenetic stimulation of the thalamus in Parkinsonian rats achieves a better therapeutic profile in terms of its improvement of motor function than conventional therapy. We're really hoping to see clear improvements in motor function, potentially improvements in sleep, and we don't want to see any adverse consequences of stimulation. And so far we haven't, but at the preclinical stage you really need to make sure of that before going forward. So in our research we are stimulating the thalamus in Parkinson's disease rats, and the kind of primary measures that we're recording are behavioural measures that index the level of motor function in those animals. So we do a number of things. So one protocol that we have established is called the skilled reaching protocol, where an animal with Parkinson's disease, we've given them Parkinson's disease by injecting a toxin into one half of the brain where the dopamine cells are and killing the dopamine cells on one half of the brain. And what this does is it leads to dysfunctional movements on the other side of the body. In the skilled reaching assay, we train the rodents before we give them the Parkinson's disease to reach for Cocoa Pops through a slot, and we count how many times they do that in 5 minutes. And when we give them Parkinson's disease symptoms, they do that much less. So they have what we call akinesia, is they have difficulty picking up their limb and grabbing those Cocoa Pops. So what we're measuring when we stimulate the thalamus is whether they grab those Cocoa Pops more frequently, whether they can use that limb better. Another thing that we are measuring in our research is EEG activity recorded over the motor cortex. The main thing that we see consistently across different motor structures in the brain in Parkinsonian animals and patients is an emergence of oscillations occurring at around 30 hertz, so high beta oscillations that occur periodically and in relation to movement. And there's a lot of literature to show that these oscillations can impair movement and they respond to different neuromodulation treatments. So what we're doing is we are using the telemeter to measure EEG activity and seeing how that changes with movement in rats and whether stimulation reduces the oscillatory activity in high beta. What we're hoping to see is that when we stimulate the Parkinsonian animals, this power around the 30 hertz or high beta range will drop off and they'll start to look more like control animals, which will tell us that the neuronal network function centered on the motor cortex is improving with stimulation. In our study, we have tested a whole bunch of different stimulation patterns. We've tested tonic patterns, which are just a regularized pulse, 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 pulse at a fixed frequency. We've tested different frequencies. And we've also tested burst patterns, which are a train of 3 or 4 pulses, and then a pause, and then another burst of pulses, and then a pause. This is one of the stimulation patterns that we've been using in our research, continuous theta burst stimulation, where you have a train of 3 short pulses. We use 2 millisecond pulses, and that train consists of 3 pulses separated by 200 milliseconds, and then it repeats over and over. And so it's just a burst off, burst off, burst off. We found that this stimulation seems to have the most consistent effects on behaviour in Parkinsonian animals, and there's a few reasons why that might be. So burst stimulation, there's some evidence that it can increase neuroplasticity, It could be that there is some dysfunctional neuroplasticity, either in the thalamus or in the motor cortex, that this type of stimulation is improving in Parkinsonian animals.